All right. Hello, everyone. So we're going to go through today gene expression. Here you have the learning objectives for this lecture, as well as these additional resources. So you have your textbook pages that you could take a look at, or the free version, the OpenStax textbook, which is in chapter 3.4 on protein synthesis. And this video. Remember, videos, sometimes they're helpful to see it in another way or hear it in another way. So this Cognito, how proteins are made, is a very good video on YouTube that you should take a look at. Okay, so what are we going to go through today? Three things. What is gene expression? Because we'll be talking about it all lecture. And then the two phases of gene expression, which are transcription, taking a DNA and forming this mRNA, and then translation, taking that mRNA and making a protein, a polypeptide chain. Okay? So what is this gene expression? Remember, when we talked about biomolecules, DNA was this polymer, right, called the nucleic acid, and it was built from many tiny nucleotides, right? That sugar phosphate backbone with the nitrogenous base. And remember, proteins, another type of biomolecule, was made up of a bunch of little amino acids, right? With the amino group, the carboxyl group. And if we use that language analogy of transcription, translation, we really see that DNA and proteins are completely different biomolecules. They're completely different languages, right? Different alphabets that are making up these little words, right? Amino acids versus nucleotides. So how does this cell translate the language of DNA into a sequence of these amino acids, right? That will ultimately fold up nicely into its functional protein. So taking that DNA and making a protein. Well, this is the central dogma that comes up over and over in biology. This is called gene expression, right? It's the process where information from a gene, a tiny piece of the DNA, right, is used in the synthesis of a functional gene product. So take a look at this DNA over here, right? This DNA will go and split open. You see this bubble opens up, right? And it will use one piece of the DNA to make this nice mRNA strand, okay? This process is called transcription. You're turning a DNA into an RNA. Specifically, it's called mRNA, messenger RNA, because it's a little messenger that will go out of the nucleus, right, and into the cell to make the proteins. Remember, the DNA is very bougie, and it doesn't want to leave the nucleus. So instead, this mRNA leaves the nucleus to go give the instructions outside of the cell. And then this mRNA that's floating around in the cytosol, that cytoplasm outside of the nucleus, will find a ribosome, maybe a ribosome that is on the endoplasmic reticulum, the ER, or maybe a freely floating ribosome. And these little factories, these ribosomes, right, will go and put together the pieces, take this mRNA and read it, like the instructions of it, to make its protein, right? You see these little green specks coming out. That's a bunch of amino acids getting joined together, right, from this ribosome. And so in the end, you get this nice protein that will then fold on itself into its kind of shape that it needs to be. And that provides the structure so that it could carry out all of its functions in the cell, right? That's called translation, because you're taking an mRNA, RNA molecule, and turning it into a protein. That's completely different biomolecules, right? You go from a nucleic acid to a protein. You're completely getting a different biomolecule. So that's like translating a language. Transcription, remember, if you're transcribing the DNA to an RNA, you're just changing a few letters, right? Those different letters between DNA and RNA, you're just having tiny little shifts. So the U and that T, you're going to be just swapping those up. Whereas translation, you're going and taking this mRNA, turning into this bigger protein. Remember, DNA contains all the information on how to build the proteins. Here you see this DNA molecule, double helix, right? And you see in dark blue gene 1, in yellow gene 2, and in green you have gene 3. Sections of the DNA will encode different proteins. Maybe gene 1 encodes a certain protein that gives you the pigment, right, in the cell. Maybe gene 2, right, goes and encodes a long microfilament, a bridge in the cell that kind of goes in a little track, a railway, right? Maybe gene 3 goes and encodes a little protein on the surface of the cell membrane, like one of those transport proteins that we've seen. So in the DNA, there's multiple genes that are found to encode proteins. They're the instructions to how to make the protein. So here, the DNA polymer made up of nucleotides have the nitrogenous bases A, T, C, and G, right? So here in blue, you see this double-stranded DNA. You have little nucleic, so these two nucleic acid strands are brought together. There's hydrogen bonding that occurs between base pairs. A and T pair up, C and G pair up, right, in this DNA. And so this DNA in transcription will turn into an rRNA, right? So with the language AUCG, right, those are the nitrogenous bases that we see, no longer a T. So in red, you see this single-stranded mRNA. So maybe gene 1, right, that little section of gene 1, you're going to go and make the mRNA transcript so that you could send it out of the cell to transcribe that specific gene. And then we're going to see later that when we undergo translation, you're taking that mRNA in the cytoplasm, right, and turning it into a protein with a bunch of these amino acids. So tryptophan, phenylalanine, right, glycine, serine, all of these, remember, there are 20 amino acids that link together and form proteins. So that's going to be our final product, all folded up nicely to do its function. We're going to see that in the mRNA, 3 little codons over here, 3 little bases form a codon, which will go and encode 1 amino acid, okay? So we'll see that later on. How can we go from that 4-letter language to the 20-letter language, right? Here we have only A's, T's, C's, and G's, and then in RNA, A, U, and C, and G. How do we go from this A, U, C, G and then form huge proteins that are so different? Well, I kind of just told you that 3 of these little nucleotides, UGG, is called a codon, right? And these 3 encode 1 type of amino acid. And we're going to take a look at the chart. You'll see that each of 3 pair codes, right, will go and form this amino acid. It's kind of like maybe the locks on the doors, right? With the keypads, if there's numbers, some doors, you punch in 3 numbers and then you can open the door, right? Well, in this case, you're going to have 3 nucleotides that will tell the ribosome, okay, this is tryptophan, right? And then you will say to the ribosome, okay, this is phenylalanine, which is 1 amino acid. GGC will tell the ribosome, this is glycine, right? And on and on. So translating that genetic language into the protein language, right, we say that triplets of nucleotide bases are the smallest units of uniform length that can code for all amino acids. This is what I just said, right? This triplet code, right, UGG, encodes an amino acid tryptophan. UUU is phenylalanine, right? So it's the genetic instructions for the polypeptide chain that are written in the DNA as a series of non-overlapping 3 nucleotide words. So basically in our language, right, this DNA and RNA language, 3 nucleotides, right, UGG, that triplet code is what kind of goes and takes one amino acid and sticks it onto the chain of growing amino acids. So these are called codons. 1 UGG, right, with 3 nucleotide bases, that's called a codon because it encodes one amino acid. And by encodes, I mean it's the instructions too. So now that we know what gene expression is, let's go on to transcription. DNA to RNA, right? So gene expression begins with our transcription. Here you'll see in the cell, it occurs in the nucleus. I told you, DNA is a bit too bougie. It doesn't want to go and, rightfully so, go and get exposed to the outside environment because mutations to the DNA wouldn't be good. Those are going to mutate the language in the DNA and then make a different protein. Remember sickle cell disease? We don't want that. So gene expression starts in the nucleus. It's important to know where transcription occurs, where translation occurs, okay? In the nucleus, this is where we take DNA and turn it to this mRNA, right? This messenger RNA. Don't worry too much about the pre-mRNA. We're going to be having this mRNA. What is it? So transcription, genetic information in the form of a DNA, is used as a template to generate a molecule of RNA, right? They're the same language of nucleic acids, except for that little U versus T. So let's go through the stages of transcription. It occurs in 3 stages in your cells. First, over here in the DNA, right, you see this molecule at the start point is called RNA polymerase, and it sits here and it initiates the start of this transcription. It opens up and you see that the DNA will unwind, right? And then you start this transcript. So this RNA polymerase will elongate the RNA transcript, the mRNA, and then termination, the little RNA, this RNA polymerase falls off the DNA, and you have a freely floating RNA transcript. That mRNA is just there. So let's go dig into these 3 steps. As I said, this RNA polymerase 2 in this case is what really matters to us. So what does RNA polymerase? Anything that is an ACE is an enzyme. Polymerase will polymerize and make a polymer of RNA, right? So it's an enzyme, ACE, that will polymerize, make an RNA, specifically our mRNA that we care about. So what does this enzyme do? It binds to a specific gene region on the chromosome. Here it's highlighted in the video. It's a start site, right? It will separate the double helix at a specific region. You see that the DNA is split open, right? Look at the DNA here. When the polymerase in green sits on this DNA, it splits open the 2. And it binds and reads the template strand in the 3' to 5' direction. So it is reading the DNA, one of the templates of the DNA that we call the template strand. It is reading it from 3' to 5', and it is creating, right? So you see that the RNA, look at the arrow here, it is going to the right. It is reading from the 3' to the 5'. And it will make the strand, that messenger RNA strand, 5' to 3'. Look at the new developing strand. This is where it started being made, right? The RNA transcript at 5', and it's adding on little nucleotides to the end of this, right? So you see this in green in the video here, you see the mRNA that's being made over here. It's formed, and this RNA polymerase is bringing in little nucleotides to build the RNA strand, right? This mRNA transcript, we call it a transcript. It's the RNA was transcribing the instructions for that gene. And that mRNA is complementary to the DNA template strand according to our base pairing rules. Remember that DNAs, they will go and have hydrogen bonds in between the bases, right? They have complementary base pairing A and T, G and C. Well, kind of the same-ish situation with the RNA. The way that the RNA knows which nucleotide to make is by looking at that template DNA strand and adding the correct bases that match the letter, right? So A will not pair with T, it'll pair with U, right? And then you're gonna have G and C that are gonna pair together. So DNA is double-stranded, and there exists 2 strands when we talk about transcription. So here you have DNA, AT is always bonded, GC is always bonded, right? When we start transcription, we open up that bubble and we start making our RNA. This is what I was talking about, is that the A's and T's will bind. And if you're making an RNA, right, and then U and A will bind, it's no longer a T. GC over here, right? So you see this RNA no longer has T's in them, right? Whereas the DNA did have a lot of T's. Now, you're going to see that the coding strand, right, and the template strand are a bit different. Let me explain to you the difference between this. The template strand, TACTAG, this one on the bottom here is the template to make the RNA. So transcription will only use one of the 2 strands as a template to make an RNA copy. It's not going to use both. It's just going to read through one. Look, the RNA here is base pairing and making this RNA based off the template. That's what a template is. Now, the other strand that is useless and isn't used is called the coding strand. Because of complementary base pairing, right? Look over here when they were together, right? ATG, ATC, right? The coding strand will have the same code as the RNA except T equals U, right? Look at the code on the coding strand, ATGA. Well, it looks almost identical to the RNA, AUGAU. So instead of a T, we have a U. So oftentimes, some people will tell you you could use the coding strand to figure out what your RNA is and just change those T's and U's. To me, that's a bit confusing. I'd rather you look at the DNA template, right? And look at that DNA template and follow it to figure out what exactly your RNA is. Just find the complementary base pairing. T matches with an A matches with a U. C matches with a G, and write it out that way. Yes, you could look at your coding strand. It's confusing because mechanistically, you're not ever going to use your coding strand, right? You could use it to check your answer, right? I do think that's good. A, T, oh, it's a U, perfect. We switch it. G, yep. A, T, oh, that's a U, we switched it. And then on and on, okay? So all that you need to know is the template is what we use to make our RNA. And then this little RNA, right, this mRNA, will then leave and unbind, and it'll go and translate into this polypeptide, right, methionine, and all these three little amino acids bonded, okay? We'll take a look at that later on. So this coding versus template strand, right? Every gene exists as a section of DNA, okay, which is always double-stranded. Only one strand is transcribed into an mRNA, right? And that's called the template strand. So the strand that is not transcribed, right, that one we say is the coding strand. And the template strand and the coding strand, those are going to be complementary, but remember, we don't really care about that, right? So let's say that this was our template strand that I gave you, right? I told you that this is the template. To find the coding strand, what the coding strand would be, all that you would need to do is really go and match base pair it together, right? So here I would say that if this was a coding strand, remember the coding strand is only, it's the other DNA piece, right? So if this was my DNA, I'd have my 5', I'd have CCG, TAA, AA, CGT, AGG, GCG, CTT, TTA, CTC, GGT, and then A, right? This would be my DNA strand that I see, double-stranded, right? This was my template. Over here in red, I put my coding strand, okay? So this above here is my coding strand. Remember, if I want to go and find the mRNA, right, I could either look at the coding strand and just change all of the T's, or I could look at my template strand and complementary base pair them. So I'm going to look at this over here, this strand, this template strand, and I'm going to go write my mRNA. There's a G over here, right? And so if I was going and making my my mRNA transcript, it would bind with a C, right? That would be my first letter of my mRNA. Then it would be C, then it would be G, then there's an A here, right? RNA polymerase will not go and put a T. It will go and put a U, right? And then the T here, you're going to say, oh, RNA polymerase doesn't care about T's. It will read the T and it will put an A. Remember, that nucleotide is fine. We do have A's for RNA, right? A. I'm going to stop here and I'm going to let you see that. Look at my coding strand, the other strand that we didn't care about. It has the same code as the mRNA, except the U. Right here, there's a T. Here, there's a U. So that's the only difference. So UAA, CGU, AGG, GCG, CUU, UUA, CTC G. Oh, here, you see I made a mistake. Here it wouldn't be a T, it would be a U, right? I was using my coding strand, GGA, okay? So this would be the process of transcription. I transcribed and made an mRNA in a mRNA form, right? My RNA polymerase would really be the one that was doing this, right? Okay, I think we're good to go. Now, let's talk about the steps of translation. So this is actually the steps of transcription, right? I think here it says translation. So let's talk about it's actually transcription. You can go make that little correction. So transcription, right? Step number 1 is called initiation. In our cells, a collection of transcription factors, right, mediate the binding of RNA polymerase. Here you see a piece of DNA, right, and a bunch of little transcription factors bind onto that DNA. Where do these transcription factors bind to? It's called a promoter. It promotes the expression of the gene. So a promoter is a non-coding DNA. That means it does not get encoded as an mRNA. It's just a place where these transcription factors, these TFs, see the little purple things, they will go and bind onto this promoter region. Okay? Several transcription factors must bind to the DNA before the RNA polymerase II can bind. So they're going to say, ooh, I see this promoter, right? And these transcription factors will help to recruit and position the gray big RNA polymerase enzyme, which will go and polymerize the RNA, make the RNA, right? So the promoter is really near the start point. You see this little band? That's the start point here. And the promoter will go and sit on this region over here, right? So first, let's go through the steps. This is step 1 of transcription. In step 1 of transcription, right, the transcription factors go and sit on the promoter. Then the transcription factors position the RNA polymerase on the DNA, ready to go, okay? So those are our first steps. I'll give you a little, just one little thing. So the promoter over here, this promoter region, right, commonly includes these TATAATs, right? We call this the TATA box, okay? It's a nucleotide sequence containing many TATA sequences, about 25 nucleotides before the start point. And these polymerases, these transcription factors first, will recognize the TATA box very quickly, right? All genes almost have this TATA box, so the transcription factors will go and bind there. Only after transcription factors are attached to this TATA box, right, does the RNA polymerase II bind to that promoter region. So you really need these transcription factors to help the RNA polymerase bind to that region. So let's look at initiation, right? So this is our transcription. Again, make sure you change that. Step number 1, initiation. Once that the transcription factors are bound to the promoter, RNA polymerase can then bind it and splits the double helix. So here are the transcription factors in our first stage of transcription. They will bind to this region, right, called the promoter, made up of a bunch of TATAs, right? It will recruit, finally, the RNA polymerase, which will then split open the double helix, right, separately. You see it in this picture over here, right? You see that it's breaking off the pairs of hydrogen bonds complementary. This is why we said there was hydrogen bonding between the nucleotides, right? We don't want very strong bonds there because we want to unzip the DNA over and over to make our genes, our mRNA, right? That will then turn into proteins. So again, transcription factors bind to the promoter region, RNA polymerase is recruited, and then your DNA is split in two. Step 2 of transcription, right, is called the elongation phase. RNA polymerase moves down the DNA template from 3' to 5', right? As RNA polymerase moves along this DNA, it continues to untwist the double helix, exposing about 10 to 20 DNA nucleotides at a time. We don't care about that. And the key here is that the 3' to 5' DNA strand that it's reading and creating its code from is called the template strand. So here, the template strand here is this one, which means that the RNA polymerase, when it starts working, it will go and make its RNA copy based off of this. And it will base pair with this template, right, to make the RNA. The coding strand will not be used by the RNA polymerase, but you're going to see that our RNA that we have matches this code very closely due to base pairing, right? So in this transcription elongation step, right, RNA polymerase, remember, is an enzyme, ACE, that makes the RNA. It synthesizes the mRNA from 5' to 3', right? Here the RNA polymerase will go through 3' to 5' along the template strand, but it will make the mRNA 5' to 3' to 5', right? So in this case, right, you're going to go and have RNA polymerase that will go and add nucleotides to that 3' end of the previous nucleotide. Take a look at this over here. You have this little nucleotide, right, with the 3' end, okay? And it will only add its next nucleotide to that 3'. So really you have this 3' to 5' addition of each of the nucleotides, and then it gets added to this end. You will never go add to the 5' end of this little nucleotide, right? So I don't think I wrote it quite clearly here. Let me kind of go over the steps. Over here, the 3' to 5' DNA strand, right? That's called our template strand. So RNA polymerase goes and along the template strand from the 3' end to the 5'. And so the transcript, though, that you're going to make, right, is going to be in the direction from 5' to 3'. You start at this 5' end and you keep adding stuff to the 3' end of your little nucleotide, right? So you keep adding 5' to 3'. So let's take a look at this in the video. It might make a bit of sense. It's going to read along from 3 to 5, right? So it'll go on and look at this 5' end of the RNA. It's adding nucleotides all the way up, right, until we get to our 3' end. So the RNA polymerase read the template and added C, G, C, A, U, G, right? Look at how they complementary base pair here. C goes with G, G goes with C, C goes with G, A goes with T. That's fine. But then T does not go with A because this is an RNA. Any T's that you put down, you turn them into U's, right? And then we get to the end of our strand, okay? This was elongation. So again, the RNA polymerase started read from 3 to 5' on the template, but created the mRNA from 5' to 3', right? it added to the little OH group, the 3' here, and created this phosphodiester bond between the two nucleotides. The last phase of transcription, step number 3, is called termination. The RNA polymerase will reach a special area called the termination sequence, and then the RNA transcript is cleaved off from the RNA polymerase, and the transcript undergoes some modifications that we won't talk about in the nucleus, and then it's ready to go, right? So then that mRNA transcript leaves the nucleus through the nuclear pores, the little spaces. Remember, we saw in the nucleus there was little pores that allowed the mRNA to leave. And now we're on to translation. So in transcription, we took a DNA and we turned it to RNA, right? There was 3 steps to that, right? Initiation, transcription factors bound on to bring and recruit the RNA polymerase. Then there was elongation, right? The 2 DNAs were split and the RNA polymerase went through, read the template, created our specific mRNA sequence. That mRNA sequence, right? Then we had step 3 was termination. The RNA polymerase went off of the little transcript, right? And it left that transcript there. Part 3 is called translation, taking an RNA molecule and turning it into a polypeptide. So in translation, right, after transcription, here we went from a DNA to an mRNA. Now we said that mRNA that's made will go and leave the cell nucleus, right? It's still in the cell, but it's going out of the nucleus into the cytosol, right? This is not outside the cell, this is in the cytosol. And we undergo translation. This mRNA language, right, will be turned into a polypeptide protein. So the language in the mRNA transcript, right, is used to make this polypeptide chain. So it's going to read through this little mRNA and make its polypeptide chain. So translation occurs coupled, sometimes it's close to the ER, right? Because the ER has the ribosomes, if it's the rough ER. So it could occur coupled to the rough ER. Or it could happen over here, like you see in this picture, just floating around in the cytosol. Many ribosomes are there. The key here for translation is you need to find a ribosome, right? To go from your RNA to your protein, you need a ribosome. Now, the genetic code for genes, right? This is a scary chart, but it's quite easy to read. There are a total of 64 codons. Remember, we said that one codon would go and make an amino acid, right? So UUU makes the amino acid phenylalanine, right? CCC will make proline, okay? So you could go look up any 3-letter code and you'll see that they go and have their little amino acid that's next to it, okay? And I know there's like some weird abbreviations, but don't worry too much about that. So we have 64 codons, but only 20 amino acids, right? Amino acids have more than one corresponding codon. This phenylalanine could be the codon UUU, and then the ribosome would bring in a phenylalanine, or UUC will also bring in phenylalanine, right? So 2 different codes will encode only one of the phenylalanine, right? If you have this code or if you have this code, you will take a phenylalanine and add it to the chain. This is because the genetic code is said to be redundant. It has these redundancies, right? Look at proline. 1, 2, 3, 4 combinations could bring in a proline, right? It's redundant. You have some different codes give the same amino acid. Why would that be, right? Well, this limits the impact of mutations. Let's say for proline, right? You have a mutation in the U in your little mRNA, or let's say your DNA changes something and the U turns into a C, right? Then you're going to say, oh my God, I got a mutation. That's bad. Well, not really. Because if you had a U and now you have the code CCC, that's still going to make proline. So this is good actually to have a bit of redundancy so that mutation doesn't always change the outcome. You'll still get a proline and you'll get a perfectly good perfectly good protein that has the same code as it should have. Yes, the DNA changed, but then that protein didn't change. This is called you don't really have any change in your mutation, right? So you have in the end a mutation that didn't make any damage. Now, the next step we're going to see, right? We're going to need to figure out how do we go from our mRNA to our protein. And the key important player is this little thing called the tRNA. We said ribosomes were important, yes. But tRNA is the little helper of the ribosome. tRNA molecules are like adapters in a way, okay? They take, let's say your new phone, right, has a stupid new charger. You could just put an adapter on it and then plug it in, right? Well, these tRNAs are a bit like it. Each tRNA molecule is approximately 80 nucleotides long. Take a look at this, ACC, ACGU, right? This is a bunch of little, what do we call it? These are a bunch of our nucleotides, right, that are all together. And it's a RNA because it has some U's, right? So this is not a mRNA because of the shape of it. This is a tRNA, okay? It's a type of RNA, but used for not as a messenger. It's used as a transfer. tRNA stands for transfer RNA. And so each tRNA molecule is approximately 80 nucleotides long and has this 3D conformation. Now, you'll see that the RNA folds in on itself, right? Remember when we had an mRNA, it was this long chain, linear? This tRNA is a weird 3D structure, right? And each contains a nucleotide triplet called an anticodon. Do you see at the bottom here? This is called the anticodon region. All the rest of it we don't care about, right? They're just base pairing into a weird way. This bottom part is crucial. 1, 2, 3 nucleotides called the anticodon is what will base pair with the mRNA. Here, in my little video, okay, you see the tRNA has a little codon that matches our mRNA that is in the ribosome. So you have the little anticodon that will match with the codon of the mRNA. Here's the mRNA codon, and it matches to the anticodon region, okay? So this anticodon is what base pairs with the complementary codon of the mRNA. At the other end of the tRNA is a specific binding site where an amino acid could be attached. Do you see these little purple things on each of the tRNA? Those are one amino acid, right? So for each codon, right, we have an amino acid. For each anticodon, right, there's a linked amino acid. So you'll see that each of these tRNA carries with it a little amino acid, okay? So let me show you how they draw it oftentimes in some books, right? This is the actual 3D structure of our tRNA. And this over here is our symbol. It looks like a little candle that we draw for a tRNA. So this symbol, right, is what we use in the book often. You have this little attachment site for your amino acid, and you have your anticodon over here. And this will bind to the mRNA. So what's the use of this, right? This is what will link and bring in the amino acid that's desired. The ribosome, right, is kind of the little factory though. Polypeptide synthesis, making a polypeptide, adding on amino acids. Look at this amino acid chain that's being formed, right? That's done by the ribosome, okay? So ribosomes facilitate the specific coupling of a tRNA. This is our tRNA. Anticodon, right? The 3 anticodons on the tRNA. It facilitates that binding to the mRNA, right? So it goes through and reads the mRNA and puts the correct tRNA on that sequence, right? Look at it. They bind the red ones to the red ones, the purple tRNAs to the purple, the blue tRNAs to the blue region, right? So ribosomes help to facilitate the coming together of an mRNA and a tRNA. If it was just freely floating, this would not happen in an efficient way, okay? And so a couple things about ribosomes. Ribosomes are made up of 2 things: a protein, because it's a type of enzyme, but also this rRNA, right? So funny enough, yes, there's tRNA, there's mRNA, but this ribosomal RNA is also found. You see protein here, and you see a bit of this RNA in the ribosome. We call that rRNA, okay? It's just that not many things are made out of RNA, and this ribosome is one of the things. A ribosome is made up of 2 subunits. A large one, here you see in blue, or here in the picture you see this huge one on top, and the small subunit on the bottom, kind of holding everything together, okay? And there's 3 sites that exist in the ribosome: the E site, the P site, and the A site, right? Those are where the tRNAs will go and bind to, okay? And we're going to go through exactly what the steps are for that. So here you see the E, P, and A, and there's these tRNA molecules are sitting in that site, and they're making this growing polypeptide. So now a ribosome structure, right, the whole point of it, the function of it, is to bring together the mRNA and the tRNA. And as I said, the large subunit, right, it contains those 3 tRNA sites, A, P, E. And they have those names for a reason. The first one, A site, is the amino acyl tRNA site. Amino, right, is the amino acid on the tRNA, right? So amino acyl tRNA, that's the site where the tRNA will go and bind. Amino acyl is acyl is like a type of bond between the amino group and the tRNA. Okay, so that tRNA will go and bind in the A site. Then the peptidyl tRNA site, right? That P site is where you're actually going to have the transfer. So you're going to be going and transferring the amino acid to the tRNA, okay? And then the E site, the exit site, that's where the tRNA, once it passed its amino acid to the chain, it will go and exit. So let's go through these steps. Translation, like transcription, has those 3 steps. Initiation, we've heard that before. Elongation, and termination. Good. They're not any different, but they're different in their mechanisms. Okay? So the first one, initiation. The small ribosomal subunit binds to mRNA. So this unit, right, this small subunit will bind the mRNA onto it. It scans the mRNA for a start codon, AUG, okay, and binds to the mRNA at that location. If we go back, way back to our little chart, AUG is called the methionine, right? That's a type of amino acid, but it's also the signal to tell the ribosome, start making the protein here. There's also stop codons that we're going to see, UAA, UAG, that means stop making this. And you see that there's no amino acid that's there. Now, let's go back. So initiation, elongation, termination. So the small ribosomal subunit binds to this and goes and reads. Then it finds AUG, right? And it binds to the mRNA at that AUG site, right? And it says, perfect. Then the initiator, right, the tRNA, binds to the mRNA at that start codon. So it always will start with a methionine. So this tRNA binds on, and methionine is the one that starts it, okay? So this is called the initiator tRNA. It's always this methionine. You can have methionines later in the chain, but this is always the one that starts it. So the initiator tRNA carries the anticodon UAC, right? Think about it. AUG is the codon, right? That was in this mRNA, but the anticodon that pairs with it is going to base pair, UAC, right? So if I ever ask you for the anticodon, you just need to think about what would base pair with A, that would be a U. What would base pair with U, that would be an A, right? U is like a T a bit, and G would be a C. So these are the base pairings, the anticodon on the tRNA and the codon on the mRNA. Now, once that first initiator methionine tRNA binds, right, then the large ribosomal subunit binds on top of it, right? The binding of the large subunit, this requires energy. It requires GTP. Who cares about GTP? It kind of sounds like ATP. That's all we need to know. You need energy to bind this on the big, large subunit. Okay, and now methionine is in this P site. Okay, elongation. The first thing that happens, right? So there's a codon recognition. So the next tRNA that needs to come in, right, recognizes the codon that's 1, 2, 3 with the anticodon on the tRNA, and it requires this GTP to do this, right? It needs some energy to go and bind there. It needs to push it onto there. So boom, right? So here, this binding requires some GTP. And then finally, you're sitting next to your other friend, right? You need to force it there a bit. Then the peptide bond is formed, right? So you see over here, this little amino acid forms a bond with this other amino acid, right? The P site and the A site, and this is called the peptidyl transfer. You're transferring a peptide bond between this amino acid and this one, the circle and the diamond, right? And so the growth of the peptide chain is linked to the new tRNA in the A site, right? This is called peptidyl transfer, right? You're going and you're transferring your bond, your amino acid chain to the single amino acid, and you've just added a new amino acid to your chain, right? And the last part is translocation, okay? So once you've passed it on here, the tRNAs are pushed forward in the ribosome. So one moves to the P site, from the P site into the E site, where it leaves the ribosome, right? So this little P tRNA goes to the E site, right? And this little A one goes into the P site, right? This requires GTP as well. And the tRNA in the A site, so you have this transfer. Now it's in the middle, right? So the E site one exits, and the P site one is now ready for the next amino acyl tRNA to come in, and then you're going to bind it on, right? So those are my steps for elongation. Let's go back. Initiation was the first codon, right? That so basically a small ribosomal subunit bound to the mRNA, searched for AUG, found it, and then put that first methionine, boom, start. Then it recruited the big subunit to come on top. Then we had our first, now we're growing our chain, right? So you're going to go and have in the A site the next anticodon on the tRNA will match to the codon on the mRNA. It'll bind, it'll do a peptide transfer, right? Because it's going to make a peptide bond with the next amino acid. And then it pushes and moves to the next site, right? And it's ready to do another next codon, right? The next three little pieces, 1, 2, 3, are now going to be in the A site ready to receive a tRNA. The last part is termination of translation, right? So elongation occurs until the stop codon arrives at the A site. So UAG, UAA, or UGA, if you go look at your codon table, you'll see that those are the little codes that you have for terminating that mRNA, right? So a protein called a release factor binds to the A site and disassembles the ribosome to stop translation. If you take a look at your codon table, I want you to go back to that table. Go look at UAG, UAA, or UGA, and you see that that code doesn't have any amino acid linked to it. That's because a protein, that release factor, binds at that UAG or UAA or UGA, and it will go and say, we're done, right? It kicks out the tRNA from the P site, kicks it out, kicks out the free polypeptide, and disassembles all of the ribosomal subcomplex, right, from the mRNA, okay? So over here, right, what happens next? So termination occurred. Now, one thing I really didn't stress enough is that the folding of a protein is crucial. If it's not folded well, we can't have our function, right? So the protein is folded in the cytoplasm or in the lumen of the rough ER. Remember, we said sometimes in the rough ER it does its little folding, and you need a proper folding to have your function. Remember sickle cell disease, we have a mutated protein, the folding doesn't work, and we're done, right? You get a disease. So here your protein is nicely folded. Again, you don't want to have fever, or else you're going to have this unfolding of your proteins, and that's not good for you. You don't function. It stays inside the cell, right? And it'll once you folded it nicely, right? It gets it could stay inside the cell and do its job inside. Here you have little proteins floating around, or it could get incorporated into the membrane, right? You could shuttle some of them and put the proteins in the membrane. Here you see the phospholipid bilayer, and you see all these other little proteins going around, right? So you really want a nicely fold of the proper sequence from the DNA to the mRNA to the protein, right? And then you want to fold it properly, and you want to get it to where it needs to go. And remember, the Golgi apparatus and a couple other organelles get it to where it wants to go. So proteins, they're synthesized from the N-terminus amino group to the C-terminus carboxyl group, right? And you're going to end up having this nice bunch of amino acids linked by peptide bonds. So let's go through these translation steps one last time with some animations, and then we'll have covered all that we need to. So step one is called initiation, right? Remember, the ribosome reads and finds the sequence AUG, right? That's the start one. So the small ribosomal subunit finds AUG, and then it's like, perfect, right? We're good. Then, once it finds that sequence, right, then remember methionine will bind on with its anticodons, UAC. And then we assemble the ribosome, right? So we're going to use GTP, this form of energy like ATP, and we'll form this unit, okay? And you see that in the P site, you now have the AUG binding to a UAC. This is methionine. When you read your little chart, right, you might need, I would provide that to you on a test, the codon chart. The codons match, of course, the codons on the mRNA and not the tRNA, right? I might ask you what the tRNA anticodon is. You could just think of what the codon would be, right, based on the chart, and you could go and find what the complementary anticodon is, right? So AUG is what matches UAC, methionine. Then, right, the actual codon, anticodon recognition, that too requires a GTP, right? So do you see how now the valine, right, came into the next spot and it bound and it did a little peptide transfer onto the next growing chain. The chain was moved to this first A site. Then we translocate. We also need a GTP at this point. And then the last E, the one in the E spot exits. E is for exit, right? And then another, you get this other tRNA that binds, transfers and elongates the chain, and then you get translocation, again, exit out our next tRNA. Another one comes in, asparagine, transfers it on, translocates with a GTP, exits the E site from the other side, and then you get your leucine binding, will form with the GTP, translocates using some energy, and then you exit from the E site, right? Now, look at this sequence here. It could have kept going on and on and on, right? But the minute you get UAA, if you go look at your codon table, what does it say? It says stop, right? That's a stop signal. That's going to be over here. Termination. It'll bind here, right? And it breaks down the translation assembly. The polypeptide will be cleaved off. You're going to have this tRNA cleaved off. The ribosome splits up and the mRNA stays floating around, right? It will eventually get degraded. So to translate the mRNA strand into an amino acid, right, you need to go through a bunch of steps. So what we're going to do here right now is we're going to do a little activity to see how that happens. So here's my mRNA sequence, right? We're going to go and make this into an amino acid, a bunch of these amino acids to make our polypeptide. So first of all, it says how many amino acids are present in the polypeptide? We don't know yet, right? We need to find first AUG to see where that is. So let's go through. If we are the ribosome reading this, right, we're going to find, over here, we have an AUG, right? So you're not going to go and find this code first. Remember that that ribosome, right, will wait until it sees its AUG before it starts. So AUG, then it'll read CCU, then it'll read ACU, GGU, UGU, UGA, right? So it'll go through these little phases. Let's go see what we have. So AUG, right, the first one it's going to make, right, is going to be a methionine. That will always be the first amino acid that you're going to have, right? Then it's going to link a CCU. CCU, if I look here, is proline. The way to read this table is the first mRNA base, you look at this letter, right? So you go to the C over here. Then you look at the second mRNA base is C over here. And then the third one, right, is U. So you're going to go look at this one over here, okay? It's just, if you just want to scan the table, you could do that too, but you could use the little sides to help you out. So CCU is proline, right? So proline. You don't need to know this chart by heart, right? Or even the names of the amino acids. ACU, so ACU, we have this this threonine. Then we have this GGU, right? So we've done 1, we've done 2, we've done 3. Now we're at our GGU. So GGU, so this is glycine, right? Over here, glycine. Okay. Then we're going to go to UGU. UGU is the cysteine, right? Then we're going to go to UGA. UGO. This is a stop codon, right? This is not going to encode. You're not going to put stop at the end. This is not any codon. So we're done. Everything will disassemble, right? And who cares if there's an A? We can even have a bunch more letters going off, right? This is going to stay as is, met, pro, threonine, glycine, cysteine, and then it's done, right? This was a stop codon. We didn't have any amino acid that got encoded, okay? So how many amino acids are present? 1, 2, 3, 4, 5, right? There's going to be 5 of these amino acids present. This, so then you're going to have all 5 of these. Here it says label the N and the C terminus. Let's go back. I'll show you. Remember that the beginning part, the amino group, is called the N terminus, and at the end you have the C terminus, right? So over here, I could write that this is the N terminus, right? And this is our little C terminus over here, right? On my protein. It's not that there's an amino acid there. Methionines NH2 group would be here, and cystine's COOH group would be at the growing end where it stopped. Okay. So the takeaways from this class is that the central dogma describes the process of gene expression, right? Genes in our DNA need to be transcribed into RNA, and this RNA must be translated into a polypeptide. Transcription happens in the nucleus of your cells, and the transcript leaves the nucleus. Once in the cytoplasm, the ribosomes bind to the transcript, beginning to build the polypeptide. And these polypeptides, they fold into functional proteins, right? So here are some questions to think about, right? So describe the details of the transcription process. Define what a genetic code is, a reading frame, a triplet, a codon. Describe in details the process of translation. In which steps do we need some GTP? And what are 3 types of RNA involved in gene expression, right? And their purpose. Just some to think about. Maybe the reading frame, this one I would cut out, not so important, okay? So that was it for our gene expression.